A very good morning, one and all present here. It's my immense pleasure she is a distinguished academician and a researcher in pharmaceutical science with over 30 years of academic and research experience, and currently serves as principal of Agalya School of Pharmacy, Palakkad, Kerala. She holds M.Pharm in Pharmacognosy and Phytochemistry and PhD in Pharmaceutical Sciences from Dr. M.G.R. Medical University. Recognized PhD supervisor, PCI inspector, NAC and NBA coordinator and journal reviewer. She has played key leadership roles in academic administration and quality assurance. Professor Dr. Srikumar has published over 86 peer-reviewed research articles, holds 5 patents, and out of among these 2 is granted, and has delivered more than 50 invited talks at national and international forums. She has received numerous prestigious awards for excellence in education and research, including Women Achiever and Women Research Awards, and actively collaborates with international institutions and industry in the areas of pharmacognosy, phytochemistry, herbal drug delivery, marine pharmacognosy, and nanotechnology. With this introduction, I welcome Dr. Sapna Srikumar for giving her talk on this today's seminar. Welcome, madam, please.
Very warm good morning to everybody gathered here. First of all, I would like to express my gratitude to the principal, management and the organizing secretary, Dr. E. Tilagam madam, who has invited me to this August gathering, and it is a great privilege for me because I have been associated with JKK Muniraja from a very long time. I was working in Sri Ramakrishna Institute of Paramedical Sciences, and from the year of 1998, I've been coming here as external, as an examiner, and the friendship that the college maintains is seen even today. So it's an honor for me to be here and to be the speaker of this wonderful seminar, which is very beautifully arranged. So dear students, good morning. Exploring. Now let us all explore. Explore is something like we see what is new, isn't it? It's like finding out what is new, what is different, what is most important. That is what we would like to explore. So I'm going to take you today to a journey where we can try to explore what is the future of herbal medicine. Future of herbal medicine mainly dealing with normal drug delivery system for targeted medicines. Traditional wisdom has been very huge in India. As you all know, everybody talks about a lot of Ayurveda drug, the uses, and worldwide, globally, it is accepted. But why it has not yet been marketed or much in use is the question, and that is where we have to set up the scientific modern techniques so that we can have formulations which are at par with that of the synthetic drugs. Unless and until that is done, we will not be able to achieve the proper medicines. So my talk will take you to a journey where we can start from the beginning, and I will try to put in some light where you can come to know as to what you can do in areas of your research, whether you are in any department, which area of research you can carry out and you can go deep into and apply herbal medicines. So are you all with me? Yes, students? Are you all with me? Fine. So, how many are M.Pharm from here? How many students are M.Pharm students? No M.Pharm students? Okay. So, let us go ahead. The importance of this topic. This topic, as we all know, is a matter of rising global interest. Why? Because more than 80% of the people rely on herbal medicines. But there are limitations also. Conventional drugs are also having limitations. The limitations of conventional drug is because it is said to be toxic, it can cause drug resistance. And that is the area where the herbal medicines come into place. But are the herbal medicines completely safe is the question that we have to ask ourselves. So in my talk, in brief, I will be talking a little bit about the introduction, key challenges in bioavailability, novel drug delivery systems, real world examples, evaluation methods, how do you carry, challenges and opportunities, future directions, and the conclusions. Can anyone tell me what are herbal medicines? Anyone student? What do you think when you talk about herbal medicines? What is the first thing that comes to your mind? Yes? First thing, when you say herbal medicine. Come on students, unless and until, yeah, please. Yeah, it is derived from plants. Right, please sit down, thank you. So, when you say it is derived from plants, it is not that it is derived from just one part, isn't it? Some medicines are only present in the root, some are present only in the leaf. So there is a variation. Apart from that, you know what is the medicinal constituent which is having the value? That is the phytoconstituent. And the plants are having millions of phytoconstituents. Millions, isn't it? So how can you find out or how can you get a single drug? Isn't it? It's very difficult. So this is the thing. Herbal medicines, when we say, we all say that it is derived from plants, it is extracted from plants. But can you make a drug which is equivalent to that of an allopathic drug? No. That much safety levels are not there. Why? Because it contains a lot of constituents and there is various levels or concentrations of the constituents which are found in the same plant if it is grown in various other places. Okay. Now, due to this complex chemistry, you have a lot of constituents and since you are separating out. But purity is also a factor that we have to keep in mind. So what are the statistics? Can you see that, do you know, how many of you know that globally there is $140 billion of market? All right, presently around $150 billion of market, which was estimated from the year of 2023 onwards. And you know what? It is going to be $500 million by 2030. In a span of 7 to 8 years, it is going to reach $500 million. Such a big hike. Now this big growth is because the newer methods or the modern techniques in the formulations, in the methods in which the herbal medicines are produced have come up. So it is very important for us to change. You have to come out of the basic part if you want to produce a medicine, because they are gems. Herbal medicines are gems. They are the diamonds which is available in the nature. But if you want to produce a proper medicine out of them, you have to know the latest matter. So that's very important. Medicines which are herbal have treated a lot of chronic diseases like cancer, TB, diabetes, inflammations, etc. And it has got a lot of activity. But the problem is in solubility, that is the bioavailability. Most of the phytoconstituents have very low absorption rate. And this absorption rate is because its solubility is very low. Understand? It is not soluble. And another problem is because of the size of the molecule. They are very large in size. The third reason is because of degradation. It gets degraded very fast when it is taken. So what happens? The dosage also you have to increase. Now scientifically I am speaking to you. You all know turmeric. Turmeric, you know. Manjal. So in manjal, what drug do you have? What is the drug? Name the chemical constituent present in turmeric. Anybody? Curcumin, isn't it? Haven't you heard? Now, if we are all telling that the turmeric is very good for health because it contains curcumin. But I will tell you the scientific fact that only 1% of what you take of curcumin is absorbed by the body. Only 1%. All right. Or even less than that. So the bioavailability is very less. Now we take the case of solubility. Anti-cancer drug. Paclitaxel. Paclitaxel is a very good anti-cancer drug. But the same problem, solubility is very poor. Okay, for paclitaxel also the solubility is less than 1%. The next one, I can give you an example of resveratrol. Resveratrol, solubility is there, but absorption is very low. Okay, it is soluble, but absorption is very low. So these are some of the examples that I am giving you to understand the statistics as to why you cannot say that the medicine that you prepare from the extracts are excellent medicines at par with what? With allopathic drugs. Understand? You cannot say that. You cannot claim it. So we have a lot of challenges, isn't it? I told you, low solubility, poor absorption, and rapid metabolism. Now what happens because of these reasons? In the stomach, what acid do you have? You have the gastric acid. Because of the gastric acid and because of the enzymes, it will get degraded very fast. And then what happens when it gets degraded? It gets eliminated and it doesn't enter your bloodstream. So these are the other problems. All right. Now imagine you are a soldier. Imagine, students, you are a soldier. Okay. You are a very good soldier, or at least you know a soldier, a person who is a very good soldier. He is very famous. The battlefield is very far and there is no transportation. Means the soldier is there, the soldier is very good, but he doesn't have the transportation to go to the battlefield. This is the condition of herbal medicines unless and until they are modified to meet the necessary things. They need to be modified. They need to be delivered in a way which is needed needed for the body to absorb. Otherwise, the dosage alone will not do. Okay. The reality. So these are all the myths that we have, that I have highlighted, the common beliefs. But the reality is that three things matter a lot. One is the dose matters of herbal drugs. Second one is there are long-time risks, so the risk factor matters. And the third one is making a formulation out of it will not have the effective result. So in order to have an effective result, you have to combat all these challenges. So now we have come to a new innovative method, and the innovative method, as you know, is drug delivery system. In drug delivery system, novel drug delivery systems where like phytosomes, lysosomes, etc., you can have a controlled effect, you can have a targeted effect, and you can have improved bioavailability, you can have improved absorption. All right, so these are the benefits if you develop medicines, or if you are in the verge of making drugs which are from novel drug delivery systems. And this is what is very important in today's life. In today's world, we do not need medicines which are of old age Ayurveda alone. It is like you are having a GPS enabled service wherein the medicines know where to go, how to go, and it will reach the destination instead of reaching somewhere else. It will reach the destination. So it is like you are having a GPS enabled service. Okay. So in short, if I have to tell, novel drug delivery systems for herbal medicines, in terms of herbal medicines I am telling, research has shown that it is precise, it has got protective effect, and it has got controlled release. Now I have done a lot of formulations on novel drug delivery systems where I have formulated phytosomes, we have made transdermal formulations, we have made gels, suppositories, as well as suspensions. And when the comparison was done of the drug with that of the raw material, when the studies were carried out, it was observed that just nanoparticle levels of the drug was enough to produce very good effect. So what is the reason now? Let us find out how the action takes place. Okay. So to understand that, we have to know at least some of the main types of novel drug delivery system. One is of the nanoparticle size, where you have the solid carriers. These solid carriers are like liposomes, phytosomes, and cubosomes, wherein the size is as less as 1 nanometer. Now when the size is as less as 1 nanometer, the benefit is that there are multiple benefits. One benefit is that the penetration through the cell takes place. The penetration through the blood-brain barrier takes place. So many drugs which do not reach the desired target, like the liver or the brain or the lungs, understand, it reaches that area. Moreover, it does not affect the healthy cells. So that's very important. So in this way, there is another modified type of formulation also, which is called as the dendrimes. Now dendrimes are, it is like a tree. Imagine you have a tree and you have a lot of gifts hanging, or a Christmas tree where you have a lot of gifts hanging from the branches. Understand? So these gifts, what I'm saying is like drugs. And these drugs can easily affect exactly the target site because there is the lock and key mechanism involved. So those type of drugs which include heavy loading and macromolecule technique are called as the dendrimes. Apart from that, you also have the polymer-based system. In polymer-based system, you can have biodegradable, and it is biodegradable and it is highly sustainable. Why? Because it uses synthetic polymers. So it becomes prepare dendrimes, nanoparticles which are of the dendrimes type. All right. So these are the things which are required to be understood. Scientifically, it has shown that, I told you about curcumin, that only 1% was absorbed, isn't it? Scientifically, it has shown that curcumin, which was loaded with nanoparticles, showed 90% tuber inhibition. That means 100% of curcumin was absorbed by the body and it has reached the site. Similarly, I informed you about resveratrol. Resveratrol has very low absorption, but resveratrol showed 5 times more absorption and it has killed the breast cancer cells. It has, scientifically, it has been proven. So there are many examples. All right. You can also prepare formulations. It is not tough. Making phytosomes or nanoparticle drug delivery system with herbal formulations is not a very difficult task. You can prepare it in your lab and there are several techniques. You can adopt any technique which you can do in order to develop it in your laboratory itself. So liposomal delivery system, this is one which is mostly used. I'll tell a little more about it. These are spherical vesicles, all right, and they act like a bubble. Imagine there is a bubble and there is a drug inside this. Fine. So it gives complete protection from any other type of gastric juice or any type of enzymes, and it can go and affect the targeted area. The importance of liposome is it can imbibe the drug which is water soluble as well as lipid soluble. So if it is soluble in water, or if it is soluble in lipid, or if it is not soluble in water also, it gets entrapped because it has both zones. Like liposomes are, if you see the particle, it will have both the ends. It will have the hydrophobic area also and the lipophobic area also. Understand? So all type of drugs can be trapped using the liposome method. In case of dendrimers, I already informed you, they are tree-like structures, and it is even more effective because it is highly branched. So since it is having a lot of branches, you can add a lot of amount in it, but still they are in nanoparticle size. That is the best part. There is one disadvantage for both, that the biodegradability, all right, in both is little high. That means it can get degradable. So in order to prevent and allow any type of drug to sustain for a longer time, you can use the polymer based system where you can increase the biodegradable polymers. Example is chitosan and PLGA. The latest which has come is the combination therapy. We all know that there are combination drugs which come like in an allopathy system, isn't it? You have combination drugs, antibiotics, but not in herbals. You don't have a combination of a synthetic drug and a herbal drug. You don't have formulations like that, isn't it? At least you must have not heard much. But the good news is that now it is possible, and they are called the hybrid drug delivery system. Hybrid means it contains both synthetic as well as the herbal medicine. What is the benefit of it? Any synthetic drug which is toxic, its toxicity can be reduced, that is the safety can be increased by adding a polyphenolic or antioxidant to it. Understand? The tiredness, the side effects due to the synthetic drugs also can be avoided. So these are the benefits of hybrid drug delivery system. These are usually used for complex disease solutions. It is like, if I tell you an example, you can imagine two different musicians playing two different instruments, and they are playing it together, and you get a new and a better tune than if they would have played alone. So similar to that, you can imagine the hybrid drug delivery system. Clinical applications, I will give some examples in cancer therapy and in tuberculosis only mainly. There are a lot of drugs which are developed, and you can develop transdermal formulations, gels, ointments, creams from nanoparticles for cancer therapy. All right. So the challenge, like if you take the drug and then it gets affected, or you have a lot of other side effects, can be easily avoided. Moreover, directly on the targeted site, it can be applied. So this is the best part. It can be, nanoparticles are cell permeable. That is, if you apply on the cell, the pore size of your epidermis is 20 nanometer, and hence below 20 nanometer or between 1 to 10 nanometer, easily the nanoparticle will penetrate and directly enter the site of action. So this will give a very safe and a highly targeted effect. Evaluation techniques. In vitro evaluation and in vivo evaluations, both we have for herbal nanoparticle systems. First is, of course, the phytochemical characterization, where particle size and zeta potential and encapsulation efficiency techniques are used. In this, you can easily come to know what is the size of the nanoparticle, all right, and how much penetration effect it has. That is measured by zeta potential. Next is the drug release profile. That means you have to do the dissolution studies and the diffusion studies. So these studies, if you carry out, that also comes under the evaluation technique, which is accepted by the Indian pharmaceutical, or it is accepted by IPC. All right. Third is by carrying out assays, like for example the cytotoxicity assays. Finding out the toxicity is actually very important, and for cancer drugs it is a must. So techniques like MTT, then safety screening techniques, there are many cell line studies. Using different cell lines you can carry out studies, and there are standard values which are available, easily available in the net. So from that the studies can be carried out. Apart from this, now in the introduction, sir had said that pharmacognosy is the origin of pharmacy. All right. Why? Because if you would see, I have touched all areas. I have touched chemistry, I have touched aesthetics, I'm touching now pharmacology. All right. So all these areas, even pharmacy practice. So the origin of pharmacognosy is very well from the pharmacy is very well from pharmacognosy. If you imagine that even for research you can get the proper roadmap, because having a proper roadmap is very important for you to get a good research work. The advanced imagining techniques you can do by fluorescence imaging, then by PET method, which is a quantitative method, MRI scanning, and bioluminescence or ultrasound technique. So these techniques are valid for carrying out the advanced imaging methods. As I said, finding out the toxicity. No medicine or no formulation is complete without carrying out the toxicity studies. Safety is priority. Even if you have a very good medicine, but if it is toxic, it is of no use. I will give you one very good example. Take the example of Adatoda Vasaka. It's a very good drug. As you know, it has been removed from the syllabus now. The reason is because the drug Vasicine is found to be toxic. So even though it is a highly beneficial drug, it is toxic. So carrying out the toxicity evaluation is very important. The different types of toxicity studies which can be done is, one is by acute toxicity. Acute toxicity means you do the simple toxicity studies by assessing of the adverse effect which is in a single drug administration. That means one person. All right, one volunteer. You have only one volunteer. So that is the first step, and it's called as the acute toxicity studies. Once acute toxicity study statistical values are satisfactory, the next step is doing the subchronic toxicity studies. In subchronic toxicity studies, you take the same dose and you apply for a larger number of volunteers. So it can vary from 28 to 90 days. That is, it's not for one day, it is increased, and for subchronic toxicity you apply for more number of people and for more number of days. All right, and it can extend till 3 months. From 28 days is minimum, and it can extend till 3 months. And then the values are calculated with reference to standards. The third one is the chronic toxicity. Chronic toxicity are long-term studies. All right. It extends minimum 6 months to a maximum of 1 year, that is 12 months. All right, so chronic toxicity studies, again you use the same dose, you don't change the dose, and with that you find out what is the level of toxicity, and then the average values are carried out. Understand? And this is also done on various patients, so the number of subjects are increased. After the chronic toxicity studies are also safe, found to be safe, it's not over. You have to do the organ toxicity. That means for which organ it is found to work on, like if it is, for example, for TB, it is for lungs, right? So on the organ, the chronic toxicity has to be carried, the organ toxicity has to be carried out. And lastly, and the most advanced, is the genotoxicity technique. In genotoxicity, you have the micronucleus assays, where you can detect if there is any DNA damage to your cells. All right, so that is very high level. Now, for simple research, the first two method, if it is done, it is very good. Like it's for an M.Pharm research, the first two one, even one single work will be having a lot of value. And then further works can be carried out. For PhD levels, you can do only toxicity. And I'll tell you students, you have a lot of value on toxicity studies. Because this is what people don't do. Nobody does the toxicity studies, isn't it? What they will do, toxicity studies, take it, apply on the hand, okay, no irritation. That is not toxicity. It is not toxicity studies at all. So it is found out that novel drug delivery system of herbal medicines has found to reduce toxicity to a very high level as compared to that of the extract. It has been scientifically proven. Understand? Because there is protective encapsulation of the drug, because there is a controlled release of the drug, and because there is precision in targeting the drug to the site where you want. So that's very important. All right. Okay, so my time is going up. I try to finish as fast as possible, but then, so there are a lot of other speakers also, and then otherwise students will get bored. So I'll just tell one thing, ma'am. Opportunities for pharmacy students. So these, as I've already told, these are the opportunities. So please hit upon the areas of research and formulation, quality control and analytic studies, regulatory affairs, clinical research, and industrial innovation of the drugs which are already, medicines which are already developed, but they have found to be toxic. I'll tell you, there are a lot of herbal medicines which have been found out every year by many, even by many multinational companies, but it is not coming in the market because they are not passing the test. The reason is because they are not developing it in nanoparticle technique. So if you do that, you can save a lot of life. It's in your hand, and it is the future of the world. Okay, it is the future of the world. Personalized medicines, smart delivery systems, all this future of the world, which is through herbal medicines. Nature provides the cure and pharmacy provides the bridge. Don't forget that. The future of the medicine is in the hand of the pharmacist. You have to do it. You are the future. Thank you very much, and I enjoyed taking, and thank you everyone.
Students, if you have any queries, you can utilize this opportunity.
questions?
Madam, you covered almost the entire carrier system for targeted delivery of medicines. But do you think it is possible to quality check, quality control of herbal medicines using the novel carrier system?
Yes, sir, quality control is possible because analytical techniques can be carried out, and there are a lot of analytical techniques like even UV visible colorimetric techniques which I myself have developed for a lot of complicated drugs, even for diterpenoids and for flavonoids. So it is possible, sir.
And another thing is we call it as when herbal drug is concerned, it's all having a synergistic effect. Having multiple number of phytoconstituents,
Yes.
having synergistic effect. So when you go for the novel drug delivery system, I think it is difficult for using crude, what currently using for treatment.
Yes, it's an excellent question, sir. This is a very good question. When you use novel drug delivery system, you cannot use the full extract. It is very important to extract the biofraction. Means you don't have to isolate the constituents. You isolate, like for example, in a plant, if it is reported that the flavonoids are having good therapeutic value, or the glycosides are having good therapeutic value, you have to extract the glycosides either by flash chromatography or by preparative TLC or by column chromatography technique. So once you have isolated the biofraction, you can easily make it into a phytosome or a nanoparticle drug delivery system. But there are challenges. Actually, that was one of my slides. Very good question, sir. I'll just tell about this. The challenges are that the drugs, that is the drugs that we isolate, they do not stay for long time. They have to be kept very safely. Understand? Because they can get degraded by temperature, moisture. So this is one factor, but we have to do research and find out techniques to combat it.
And with this, I would like to conclude that about the madam's presentation, we as a pharmacognosist, we understand that as a pharmacognosist, we are proud that pharmacognosy is the base for all medicines. So in that cases, so it is a base. We are facing so many challenges. That challenges is well explained by the madam, and like solubility issues, biocompatibility issues, and delivery system issues. There are so many challenges, and if at all wanted to go for novel drug delivery, targeted delivery, and one thing I understand that we have to go for the pure molecules like curcumin and
Pure biofraction.
Refined fractions, we can say.
Refined fraction is enough.
Yeah, refined. Almost we have to go for the refined fractions. And the overall presentation is, it's a right time and real time demand for the herbal medicines. Right? Thank you, madam. It's a wonderful presentation. You covered the entire thing for novel drug delivery for the our herbal medicines. Thank you very much, madam.
Thank you very much.
And I thank our organizing secretary. Thank you, madam, for this opportunity given to chair the session. Thank you very much.
Thank you, ma'am and sir. Now I request the Vice Principal, Dr. E. Tilagam ma'am, to honor our speaker, Dr. Sapna Sri Kumar ma'am.
படிப்பு சரி கம கார் படிப்பு சரி Sarigama Karma.
I request Dr. P. Satish Kumar sir to honor our chairperson Dr. N. Adirajan sir.
I request Venkatachalam sir to honor our chairperson. Thank you, sirs. Next, we will start second session. It's my pleasure to introduce our second session's chairperson, Dr. V.R. Ravikumar. Dr. V.R. Ravikumar is a professor and head of the Department of Pharmacognosy in Erode College of Pharmacy at Erode, with over 25 years of teaching and research experience. He holds a PhD in pharmacy from Sastra University, Tanjavur, along with M.Pharm in the Department of Pharmacognosy and B.Pharm degree from the Tamil Nadu Dr. M.G.R. Medical University. Now I request Dr. V.R. Ravikumar sir to introduce our today's second speaker, Dr. S. Jubi ma'am.
So, respected organizer, Dr. Tilagam, and all other coordinators of this functions, faculty members of ANNAI JKK, so Muniraja College of Pharmacy. So, this session's speaker, Dr. Jubi Madam, Associate Professor, Department of Pharmaceutical Chemistry, and delegates from various institutions. So, good morning to all. So now, I like to introduce eminent speakers. Dr. S. Jubi, M.Pharm PhD, Associate Professor, Department of Pharmaceutical Chemistry, JSS College of Pharmacy, OT, JSS Academy of Higher Education and Research, Mysore. Dr. S. Jupi is distinguished academician and a researcher with over 23 years of the experience in the pharmaceutical education and research. She earned her B.Pharm from SP College of Pharmacy in the year 1999, followed by M.Pharm from KM College of Pharmacy in the year 2002, where she was recognized as the best outgoing student, and completed her PhD in 2014 from JSS Academy of Higher Education and Research, Mysore. Her primary research interests focus on the discovery of novel and chemical entities of the cancer therapy, particularly through in the in silico tools and drug resposing. And Dr. Jupi has made a significant scholarly contribution with 105 international and 35 national publications in the reputed index journals, according a cumulative impact factor of 212.62. Her scholarly reach includes 2,512 citation with I-index of 24, means Google Scholar, 20 Scopus and 16 Web of Science. Dr. Jupi has granted 7 Indian patents and has published 12 more, in addition to authoring 5 books and 9 book chapters. Her active research is supported by multiple grants and including AACT, RPS, ICMR and JSS AHER, current projects as a principal investigator. DST as a co-invigilator in 2 projects. International grant from Sahara University, Saudi Arabia. 5 institutional grant from JSS Agha as principal and co-invigilators. She is also president of DST and ICMR travel grants from Government of India. In her academic service, Dr. Jupi plays an instrumental role as an academic coordinator of the college. She has contributed significantly in the institutional quality and engagement through her involvement in the NAC, ACPE, NBA and NIRRF. A passionate mentor, she has successfully granted 6 PhD scholar, 15 M.Pharm and 15 Bachelor of Pharmacy students with 7 PhD candidates currently under her supervisions. She has participated in 19 international and 42 national conferences and has served as the invited speaker at the 2 international, 26 national conference and 28 webinar. Dr. Jupi holds life membership of leading professional bodies including IPA, APDI, APP, SPTA and IFERP, and Associate Fellow of the Antiviral Research Society, India. She has also organized numerous national conferences, webinars and health awareness program which supported from the government agencies. Her outstanding contributions have earned her several prestigious accolades, means of Pharmacy Woman of the Year 2023 APTA Central, Pharmacy Woman Teacher Award 2023 APTA Tamil Nadu, Young Research Award 2019, Young Performer of the Year 2020 APP, Best Research Award 2019, Research Excellence Award 2025 JSS Agha, Best Teacher Award 2022 JSS College of Pharmacy OT. Her profile has been featured in famous Indian nation Who's Who. A firm believer in the wisdom of the Buddha, the mind is everything. What do you think? You become a Dr. Jupi is no deeply committed to nurturing competent and committed and compositional pharmacy graduates. So now I invite very familiar the resource persons, GSS brand, Dr. Juby Madam, to present over here the topic for now. Phaceous americana means agagoda plants. Agagoda plants, so it contains especially fatty acid is there, oleic fatty acid is there, especially for rheumatoid arthritis. Please welcome Madam to give the very pleasant presentation over here. Thank you Madam, thank you very much for giving the chance to me. Fine.
Thank you sir for your nice introduction. So good afternoon students. Yeah, I know you are anxiously waiting for your lunch, okay? Yeah, so I make your job and I make you comfortable for the next 35 to 40 minutes, okay? So we make a deal, correct? So that 30 to 45 minutes, you can use your mobile phones, but what I am telling, you have to use that. Okay, I will be telling some databases, some websites, so that you should use it. Or you just make a pic, okay? That pic after going home, no, you can use it. Okay, the deal is fine? Yeah. So a very good afternoon to one and all. So at the outset, I thank the principal, management of JKKM College of Pharmacy, especially the organizer, Tilagam Madam, for giving me an opportunity. Okay, so I will be talking about the network pharmacology. Okay, a network pharmacology and how the network pharmacology can be effectively used for exploring a plant which is called Persea americana. Okay, we call it as avocado. I think in Tamil we tell jelly fruit, correct? I think I can speak Tamil also in between. Jelly fruit, kelipattirukingla pa? Jelly fruit, green color la irukum. Okay, so how effectively you can use it. Okay, so ma'am has given the outline how the phytopharmaceuticals can be effectively used in various aspects. Especially she thrown light on the various drug delivery systems. Okay, starting with nanoparticles, liposomes, all. Now my delivery will be the drug discovery approach. Of course, I am a medicinal chemist. Okay, so basically we deal with some softwares. Okay, yeah. So let me begin. Slide change. Slide change. Yeah. So quickly moving for the introduction part for just 10 minutes. Okay, then directly we'll be going to the case study. So I think you might be knowing about the drug discovery. The drug discovery process. I understand many of you are from UG third year and final year. Okay. So the drug discovery, no, before 1980s, it all depends on the accidental drug discovery. Okay, the very good example is Alexander Fleming's discovery of penicillins. Correct? He never know that he will invent penicillin. He was a microbiologist. Correct? So it was an accidental discovery. Then later on, people went on synthesizing or they went on isolating the molecules without knowing whether these compounds will give activity or not. That we call it as random screening. Correct? That was before 1980s. Later on, then we have moved on to one drug, one target approach. That we call it as magic bullet approach. Correct? So you are giving a medicine, that drug, or you are giving a molecule. That molecule should find out the particular organism which is present in our body and it should kill. We call it as what? The basics of chemotherapy. That is magic bullet approach. One drug, one target approach. All our drugs, what whatever it is in the market, you take isoniazid, metronidazole, all the antibiotics, they all will be targeting. Okay, so the target is very important. Target is nothing but it is a biomacromolecule which is involved in the particular disease. If you take HIV, okay, so the protease, HIV protease is a target, or anything. If you take a disease, a particular macromolecule which is involved. Now what I am going to talk, what is the present era? The broad spectrum is multi-targets. Okay, so a disease, it is not defined to a single target. I need your attention here. It involves, okay, it is involved or it involves so many genes or so many proteins which is involved in a particular disease. So that's what the multi-target approach, that is a broad spectrum. Okay. Yeah, so what is network pharmacology? A new discipline, it is network pharmacology, which attempts to identify drug actions and interactions with multiple targets. So here you will be integrating the polypharmacology and the computational approaches, the tools, the softwares. Normally you use in your mobile phones, no any apps. Likewise, you will be using so many computational tools to integrate or discover some molecules. That's what the network pharmacology. So we'll be integrating polypharmacology and computational power. Let me tell what is polypharmacology. So before that, for all these you need data. I think now everywhere you can go, you can hear two words, correct? AI and ML, correct, no? Artificial intelligence and machine learning. For all you need a data. For all you need a data. So big data you should have. From that you can derive some algorithms. By doing that algorithms you can discover some new molecules. That's what it all about. So you need the big data. I told polypharmacology. So it will go both, like travel together. Network pharmacology and polypharmacology. Okay, it's very simple. So I told already it is a broad spectrum and you will be talking about multiple genes or multiple targets. So that is polypharmacology. The polypharmacology, it is a field of study related to design, discovery, synthesis and use of pharmaceutical agents, very importantly, that act on multiple targets. Always you keep in your sulfonamides pathway. Sulfonamides, correct? You start with para-amino benzoic acid. You have a pathway. We teachers, we always tell you go through this, correct? Dihydrofolic acid, tetrahydrofolic acid. You have so many inhibitors at that particular pathway, correct? Sulfonamides will act, then you have folate reductase inhibitors, correct? So what I am trying to tell here, in the same pathway, the molecule should act against multiple targets, correct? Multiple genes. So that is polypharmacology. Okay, so what does network pharmacology do? Why you can think it of why this madam is again using that word network pharmacology. What it is basically? So basically you have the data sets. Okay, I told already you should have the database, correct? The database, no, it may be a drug bank, it may be a protein data bank. Okay, how we are depositing our money into the bank. Similarly, biologically you have some databases, protein database, you have drug bank. All the details details of the drugs will be fed. The details of proteins will be fed into the protein data bank. Likewise, you have the genes database, you have the drug database. From that, you have to retrieve the data. You can take what you want. Okay? Then afterwards, you do the network analysis. The network is very simple. In this room, we are sitting around 500 people. We are in the network. We are in the network. Each and every people know we are, imagine we are a gene. Each and every person is a gene. We have some connections. We have some connections, correct? The person, those who are sitting very close to you, okay? You have more connection, correct? உன் பக்கத்துல உட்கார்ந்திருக்கவங்க கிட்ட உனக்கு ரொம்ப கனெக்ஷன் இருக்கும். நல்லா நீங்க பேசலாம் எல்லாம் பண்ணலாம். But those who are sitting in the last row, you have less connection. Likewise, biologically, the genes are also in connection for a particular disease. That's what this. Okay? You do the network analysis, then you have to validate is very important because anybody can click, you will get the result. As a pharmacist, you should know the interpretation. How far you are going to take it further. Yeah. Now, I want your attention here. So this is the outcome of network pharmacology. You will be identifying the hub genes. The hub genes. Always, I used to tell my students, always you imagine hero and heroines. What are the important genes? Okay, so at the end of the network pharmacology, you will be finding out the hub genes. Hub genes, they control the pathway, disease pathway. They control the particular disease. That is hub gene. Okay, you will be identifying. Okay, then you will be getting examples. I will tell some example. P53. Okay, it is a tumor suppressor gene. Normally we have, everyone we have this gene, tumor suppressor gene. The cancer patients, the P53 gene level will be less or it will be mutated. That is, if there is any defect in this gene, then only the disease will progress. That means the P53 is a very important gene. It is a hub gene. Okay, likewise, I have given some examples. Okay, so for MYC, AKT, TNF, okay, so these are the important hub genes. Then, this is, I need your attention. This is a typical network. Okay, you will be having nodes and edges. I have given our own example, correct? The node is each and every person. We are all the nodes. And the edges, the connections. Okay, the lines which connect each and every node. Okay, so that is edges. So here, this dotted lines, the lines are edges and this rounds are nodes. In a network, you keep, imagine the rounds are genes and the lines are edges, the connection between 2 or 3 genes. Okay, and yeah, this is one particular network. You can see here for your understanding, I have given an example. Okay, so all the genes name you can see here. Okay, it is MMP9, PTGS2, ALB. So these are the different genes and the lines. Lines are connecting between each and every genes. This is a typical display for a biological network. I think, are you all with me? Hope you are getting, correct? Yeah. Next, nodes and edges are over, and for each and every experiment, we need some results, correct? If you do the assay, what you will observe? You observe the titer value, correct? You observe the endpoint and you do some calculation. Likewise, for this network analysis also, you have some parameters. we have some parameters which tell you whether this network is good or bad, whether how much connections each and every gene has got. Okay, so you have 3 parameters. You have so many n number of parameters, but I am going to teach you 3 parameters. The first one is degree, the second one is closeness, and third one is betweenness. Degree means each and every gene, how many connections it has made. For easy example, I will tell you, in this conference, each and every person is a node. How many friendships, how many connections you are making? Okay? So the person who is making more friendships or more connections means he has got more degree. Likewise, the gene which has got more connections is having more degree. Got it? You got the point? Then the second one is betweenness centrality. Betweenness centrality means how closely it can make the connection. Okay, betweenness, how often a node lies on the shortest path. That means, for your easy understanding, I will tell, a person who is sitting in the center, the SAR who is sitting in the center, he is very close to everyone. Like that, it is betweenness. And finally, the closeness. The closeness is the average distance from a node to all other nodes. How quickly? E-road is a very center place. It connects so many places in and around E-road. So that is closeness. So these 3 are very important parameters: degree, betweenness, and closeness. Okay, and we have seen the nodes and edges. Yeah, this is one particular example you can see here. So these are the different nodes, and these are the degrees, and these are the betweenness and closeness. Okay, so now with this applications, I am not talking, or I am not going to read it, just applications. Where and all you can apply this network pharmacology. You can apply for analysis, comprehensive analysis in the drug target interactions. So that's what all about, no? We are all towards the discovery of new molecules, or exploring the plans for the discovery for some disease. The second application is rational drug discovery using computational models. Then you can identify novel targets. That's what I am giving you message for all the students. There is some disease, but there may be one target which is not explored. இதுவரைக்கும் யாருமே கண்டுபிடிக்காத ஒரு டார்கெட்டை you can identify. So that is very important. Then repurposing opportunities. Okay, ma'am, I am not going to find. Oh, no problem. You can take the existing drugs. There are so many drugs. You can go for repurposing, drug repurposing opportunities. Minoxidil. Minoxidil is an antihypertensive drug. Now it is used for the treatment of hair loss. People are using. It is a blockbuster drug. Correct? Likewise, you can go for drug repurposing and data-driven prioritization. Okay? You have 100 molecules in your hand. Of course, you cannot go for all the 100 molecules. You can go for the prioritization. Yeah, so with this, I will be moving to the case study next 15 minutes. I think our deal is fine. It is working, correct? Yeah. Now we can explore the various databases. So this network ethnopharmacology means how you can explore or you can integrate the plants, the various plants in this network pharmacology. So that's what this ethnopharmacology. Yeah. So you can see my title. What is my title? Exploring the potential bioactive constituents from Persea americana. Okay. So I have taken a example, Persea americana, how it can be explored for the treatment of RA, rheumatoid arthritis. Okay, so very quick introduction about the disease. I think you might be knowing, especially those who are elderly persons at your home, they will be suffering from this disease, rheumatoid arthritis, especially the disease which affects the joints, knees, okay, and it will be became inflamed. It becomes redness, okay, especially the joints, okay. You can ask me if there is no treatment for this disease. You have treatments. All the treatments are symptomatic treatment, symptoms based, like how we are taking the drugs for normal fever in what I can say, paracetamol, aspirin, ibuprofen, okay. And the another type is, they will be called disease modifying anti-rheumatic agents. There is no direct treatment for this, okay, and it is autoimmune disorder. So we have chosen the plant Persea americana. This is the plant. I told in Tamil we call it as jelly fruit. Correct appa? Paathirukingla illaya and the fruit? Have you seen this fruit? Yeah. So this is basically, especially in Western countries, Northern America and all, they used to treat for this inflammatory diseases, like our grandma remedy. Correct? The moment when we have any burns at our home, immediately we go and apply turmeric. Correct? Ma'am, morning she told. Curcumin. Curcumin is an important constituent of. Likewise, people are using this particular plant for the treatment of RA in this Western countries. So that's why we have selected this plant. Okay? So literature review is very important. That's why the guest of honor, Professor Manimaran sir, he told exactly, you have to go for a proper literature review. Okay, then we have chosen this plant. So this is the research gap, why we have chosen this plant. And I told no, multiple targets, multiple pathways. This is a typical example. You can witness here. If you take RA, rheumatoid arthritis, there are so many pathways involved, starting with Notch, SVK, P13, JAK signaling pathway, whatnot. Like so many targets and so many genes are involved. So this is a complex disease. Okay? Yeah, now we are coming to the tools. So these are all the tools what we have used for this study. All are publicly available. We didn't pay a single rupee. All are publicly available. So the first one is impact. Impact is to collect the constituents of the Persea americana. Then Open Babel, Data Warrior. It is a database for checking the ADMET. Then there is a database called BindingDB. Then there is a database called DisGeNet, Dvenny, Cytoscape, Pyrex. Pyrex we have used for the docking study. Then Discovery Studio Visualizer we have used for the visualization of the interactions. So this is the first phase. Okay? In another 10 minutes, 10 to 15 minutes, can I have your attention? Yeah. So the first phase, what we did, we have gone for the impact database. Okay, we fed into the Persea americana and we have collected the constituents. Then we have predicted their targets. Then we did the same thing. We have the disease. Okay, what is the disease of our interest? What is the disease of our interest? RA. RA is nothing but rheumatoid arthritis. And what is our plant? Plant Persea americana. PA is our plant, RA is our disease. Okay. So we collected the target targets which are involved in the RA, rheumatoid arthritis. Then we did the network analysis. So quickly we will be moving. So the first database, even you can take the picture or whatever it is. What database it is? Impact database. It is nothing but Indian medicinal plants, phytochemistry and therapeutics. The moment just you type the plant of your own interest. Of course, I am interested in this Persea americana. Your plant or your interest will be different. You can type the plant, you will get the constituents. Okay? And yeah, same where we have typed the plant and we got the constituents. And for the database is, you have to convert the molecules from one format to another format. Correct, no? How we are preparing ourselves for coming to college, correct? Or if you are going for a function, you will prepare something differently. Likewise, for the system suitability, for the software suitability, you have to convert the format of the structures. Similar way here, we have converted mole format to STF format. Okay, for that we have used Open Babel software. Okay, then ADME is very important. Okay, ma'am told rightly, toxicity prediction, safety prediction is very important in plant constituent aspects. You can go for ADME prediction in silico level using software like solubility, partition coefficient, okay, various parameters, ionization parameter, all these things. So likewise, we did all the ADME, okay. So the constituents collected were from the fruits and from the leaves. Okay, we have collected from the leaves and from the fruits. Okay, now next part is the ADME properties. That's what I told here. You have the limits. Always you have the limits. Your constituents, your properties of the constituent should be within the limits. Okay, then very importantly, target prediction. So we chosen the plant, we collected the constituents, correct? We checked the ADMET properties also. Now what is our next duty? We have to identify the suitable targets. For that also we have a database which is called binding DB, which is called binding database. Okay, so here you have to keep your, you have to input, you have to load your plant constituent. It will give you the targets. It will give you the targets. Okay, like celecoxib means COX-2. We are telling, no? Correct? Dihydrofolate reductase inhibitor means trimethoprim. Or in other way, trimethoprim target against dihydrofolate reductase. Likewise, each and every constituent, it has some targets. So that you can find out by using this binding DB database. So what we did, we have a cutoff score. We have a cutoff score 0.5. The score which is more than 0.5. So that targets and all we have collected. All those targets we have collected. So 25 constituents from the leaf and 4 of these constituents from the fruit. Okay, so these are the constituents and we have collected. So how many we have got? We have got 499 genes. Okay, 499 genes have been collected for the plant and leaf constituents, and out of these 499 genes there are repetitions. So deletions, after deleting all the repetitions, we have got 209 genes for the plant. So we finished one work, we have got 209 genes for the plant. And I told RA, okay, rheumatoid arthritis. For the rheumatoid arthritis, what are all the genes involved?
What are all the genes involved? So for that we have used another database which is called DisGeneNet. The name itself is very clear. Disease associated genes. We have gone for DisGeneNet. Got it? So this DisGeneNet has given 2723 genes which are involved in the rheumatoid arthritis. Correct? Now, very basic mathematics. Our school mathematics, we went for Venn diagram. We went for Venn diagram for what? To find out the common genes. To find out the common genes which are associated in the particular disease and plant. Okay? So we went for the Venn diagram. You can see here 86 genes. There are 86 genes. I need your attention here. These 86 genes are common for both Persea americana as well as rheumatoid arthritis. So this 86 genes only we are going to construct the network. We are going to construct the network. Okay, so again the Cytoscape software you can download it in your laptops and you can install the software. It's very user friendly. You have so many YouTube videos. My own videos are also available for this Cytoscape. You can check it and you can easily you can go for this steps. So this is the typical network. Among the 86 genes, these are the top genes. These are the top genes like MMP9, EGFR, VEGF. These are the genes which are closely associated with the disease. Okay? And yeah, you can recollect it before the during the introduction part. I told these are the three parameters: degree, betweenness and closeness. Correct? So these are the degree, betweenness and closeness. Okay? And the gene's name, vascular endothelial growth factor, it is coming as first. Okay? VEGF. And this is the table which gives you the degree, closeness and betweenness. Same thing. And yeah, now the first one is the PPI network, the protein interaction network. Now I have to find out the constituent from the plant. So that is PDI network. So what I did, I did the network with the selected genes, the top genes. I told one word, hub genes. Can you remember? I told to relate with hero and heroines, correct? Hub genes. So that hub genes, we did the network with the plant constituents. Okay? So the luteolin has got highest degree. That means it has got more, 124. More connection it has got. Okay? So we have, this is a typical network with the plant constituents as well as the avocado. That is, then finally we didn't stop there. Even though we found luteolin as a top drug, again we went for the structure based drug design. That is molecular docking and dynamics. We went for that. So we did the network construction, then we did the structure-based drug design docking by using Pyrex, and we found out the interactions also. Okay, very important quantitative and qualitative interactions. So we did the molecular docking study. Okay, and I think for want of time, I am not going to explain the steps. You have defined steps for this molecular docking study. You have protein preparation, ligand preparations, okay? Then you have to generate a grid, receptor grid generation. So all these things you have. And yeah, this is the analysis, and you have the binding affinity. I think you might be knowing about the score, docking score. It is nothing but the binding affinity, the interaction energy, basically it is. Okay? So again, luteolin, the first target is PTG
PTGS2, prostaglandin endoperoxide synthase 2. This is the top gene which has come out of this molecular docking study. Okay, and this is the interactions. Always there is a quantitative is your score, your binding energy, and your qualitative is your binding interactions. Always you have to see. Yeah, so coming to the conclusion, based on this study, we have developed a novel hypothesis. Okay, what is our novel hypothesis? We have found out the luteolin and the gene is PTGS2 gene. Okay, so we know very well this PTGS2 gene. This gene is very important, okay, for it is involved in the inflammation. It activates PGE2, okay, so you will get the inflammation. It is a very important gene which is involved in the pathway. So, students, my message is here. You can use so many softwares, so many databases, okay. It may be of any plant of your own interest. It is not only avocado, it is not only rheumatoid arthritis. It may be any disease of your own interest, any plant of your own interest, okay. After developing, this is the conclusion. We identified the PTGS2 gene as well as the plant constituent is luteolin. Okay, so now this is very important. You can, some of you maybe from pharmaceutics background, some of you from pharmaceutical analysis, or some of you from chemistry, what you can do with this network pharmacology. Okay, so the people, those who are very good in silico design, I am very good, I am comfortable in front of sitting the system, I am not doing any lab work. You can take that work to further. You can do molecular dynamics, you can do DFT studies, okay, binding energy calculations, all those things. Those who are very good in formulations, I am very good in formulation. You can select that particular drug and you can go for various formulations according to your own need. It may be a conventional delivery or it may be a novel drug delivery. And for the people, those who are very good in synthesis, you can find out the drug by network pharmacology and you can go for the modifications. How? From the salicylic acid only people have derived aspirin. It is an analog design. Similar way, you can synthesize your own compounds. Okay? And the people who are from pharmaceutical analysis background, you can go for the method development for the quantification. Okay? And finally, for the people for pharmacology, you can, of course, I am telling you one more message. Always you have to prove your in silico results with in vitro results. Otherwise, the people won't believe. Correct? You have to do. I have found out luteolin. Again, the luteolin has to be proved in vitro scenario. Okay, so the people, those who are from pharmacology background, they can go for the in vitro evaluation. Okay, so with this, these are the references, okay, for the study, what we have used it, and the same work has been published. It is also publicly available in current rheumatology reviews. Current rheumatology reviews, you can check out, all the details are available and all the databases are freely available. Maximum they will ask for a license, academic license. You can give you a college email ID and you can get the details, okay? and acknowledgement. So I am coming from JSS Academy of Higher Education and Research. So my humble pranams to His Holiness Swamiji. He is the Chancellor of our whole university, and my thanks to the leadership of JSS Academy of Higher Education and Research, and especially the Pro-Chancellor, Dr. B. Suresh sir, Vice Chancellor, Registrar, and my Principal, Dr. S.P. Dhanapal sir, all, and my sincere thanks to all my students. So this particular work is done by one of my B.Pharm final year students. Okay, like you, they are also interested. They learned all the softwares and they did the work and they published also. Likewise, you have to do with this conference, at least you should learn something and go back. It should not be a one another conference, correct? Because the organizers have taken lot of efforts. We have come on all the way, we speakers. So you should learn something and you should be a responsible persons for this nation. Okay. So with this, once again, I thank the organizers, the principal, and all the convener, Tilaga madam, and all my teacher friends for giving me an opportunity. Thank you. Thank you all.
Thank you, madam, for your wonderful session. So, my dear students, so keep quiet. You know, last 1 hour observing here, no, will be making noise. So keep quiet, please. So, see, we have here wonderful personalities. Here, Dr. Sanmurajan is there. Your JKK Muniraja College's alumni, first batch students of this college. Am I right, sir? Alumni of this college. Then our friend, Kandasamy is there, President APTA. Then here, Sri Kumar, and first session, the principal of College of Pharmacy, Kerala, Sri Kumar is there. Here, a lot of wonderful personalities there. So we have to listen, seriously you have to listen, what are the, research work that people, resource person presenting here. Dr. GP madam is, I think, very great. That butterfruit, very famous in the Ooty. I think the butterfruit and avocado same, no? Butterfruit and avocado, both are same only. So when we are, Sanmurajan, myself, when I study in the Ooty, so whenever will be come down here in the plain place, will be purchase avocado means butterfruit. They'll be make the juice. If you make no juice along with the milk, it'll be looking like buttermilk. It'll be very thick and buttermilk. So because of its contains no fatty acid is there, especially oleic acid is there, it'll be used for treatment of rheumatoid arthritis. So madam is very clearly explained about its chemistry and through the docking source, full dark scores. It'll be indicates it has a full of the rheumatoid arthritis. Madam is yesterday told that you are the chairperson for second session. Suddenly I'll be searched that Pessaria Americana. I collect some important and key points. It will be specially used for arthritis. Means அதாவது பார்த்தீங்கன்னா ஹிப் பெயின் பார்த்தீங்கன்னா ஜாயின் அதாவது இடுப்பு வலிக்கு வந்து இது ரொம்ப யூஸ்ஃபுல்லாக இருக்குது. அண்ட் ஒன் மோர் இம்பார்ட்டன்ட் கேரக்டர்னோ இல் பி யூஸ்டு ஃபார் த செல் ரீஜென்ரேஷன்ஸ் அண்ட் ஆஸ் வெல் அஸ் தோஸ் ஆர் சஃபர் வித் டெங்கு ஃபீவர். டெங்கு ஃபீவரில் சப்பரேட்டாக பார்த்தீங்கன்னா கால் வலி மூட்டு வலி ரொம்ப இஷ்யூஸ் நிறையா வரும். அதுக்கு if you regularly take the avocado juice, maybe in the aqueous form, otherwise the isolated form, it will be wonderful remedy for all type of the arthritis. Okay, so in the right time, madam, Jupi madam is given and right research basement for you guys. In future, you may formulate different dosage forms. Maybe for rheumatoid arthritis, so you may find the spray form, otherwise ointment form. So whatever the dosage form available in the area of the chutis, you may formulate. Okay, you may formulate. You have the great opportunities. Okay, got the Jupi madam as a resource person for these sessions. So if you have any doubt regarding this avocado plant, you may interact with our great resource person, Jupi madam. I know very well about Jupi madam. Last 25 years, it's a very eminent, very active personality. Okay, almost will be concentrate. Research work. The JSS JSS Higher. It's a great placement to do the research for research area of the pharmaceutical science. Thank you very much, madam. You may interact, please. In the session account, you may interact. If you have any doubt, you may clarify with our great resource person now. Okay, thank you, madam. Thank you very much for present over here, for wonderful assistant. Thank you very much, madam. Thank you for all our, organizers of the college.
Thank you, sir.
Student and as well as teacher organizer. Thank you, madam. Thank you.
Now I request Dr. Sumati ma'am to honor our speaker, Dr. S. Jubi ma'am. I request our Dr. Suresh sir, HOD in the Department of Pharmacology, to honor our chairperson, Dr. V.R. Ravikumar sir.
We will start the third session. Now I introduce about Dr. C.S. Kandasamy is a senior academician in pharmaceutical sciences with 28 years of combined teaching, research and administrative experience. He is currently serving as a vice principal at ANNAI College of Pharmacy, Dindigul. He has published 9 international and 20 national research papers, guided several postgraduate and undergraduate research projects, and actively contributed to conferences as an invited speaker and coordinator. He also serves in leadership roles in professional bodies such as APTA of ANNAI, All Pharmacognosists and Phytochemical Association. Welcome you, sir. I welcome Dr. C.S. Kandasamy, sir, to introduce today's third speaker, Dr. T.S. Shanmugarajan, Professor, Department of Pharmaceutics, Wales Institute of Science and Technology, Advanced Studies, Chennai.
A very good afternoon to all present here. I am indeed very happy and immensely pleased in introducing the speaker, the resource person, an eminent personality, a role model to most of the younger generations in the research. I take this opportunity to introduce Dr. T.S. Shanmugarajan, an eminent alumni of JKK Muniraja College of Pharmacy. He is currently the professor in the Department of Pharmaceutics, School of Pharmaceutical Sciences, Vistas World's University, Chennai. He holds around 23 years of academic and research experience. He holds B.Pharm, M.Pharm in pharmaceutical biotechnology and PhD degrees, and has an extensive expertise in meningeal stem cell differentiation, regenerative medicine, and advanced drug delivery systems. Dr. Shanmugarajan has received several prestigious recognitions, including the Best Teacher Award from the Association of Pharmacy Professionals, Indo-US Convention 2021. Multiple Research Faculty Excellence Award from Vistas and accolades for high impact SCI and JCR publications. He has delivered invited lectures at national and international forums, organized major conferences, and holds 2 granted patents in topical emulgel scaffolds and breast cancer diagnostic instrumentation. He has successfully guided numerous PhD postgraduates and undergraduate students and has published extensively with a strong cumulative impact factor in high index journals. His ongoing funded research focuses on hydrogel and nanofiber based regenerative therapies for bone, skin and ophthalmic preparations. Participants, delegates from various parts of the globe, kindly make use of the valuable session to be presented by the resource person, Dr. T.R. Shanmugarajan, a well-experienced expertise in the field of pharmaceutical sciences all over the globe. He has traveled a long distance. Kindly be silent, give a way for others to make use of this session. You can take a lead of this particular work in your future studies. So with this short introduction, I invite Dr. T.R. Shanmugarajan sir to deliver a lecture. Welcome you, sir.
So thank you very much, sir, for your brief introduction. And now it is a very big task for me because this session, after this session, you have to go for lunch. But anyway, I would like to explain in a simplified manner. Okay, first understanding is very important. So first I have to thanks to the management, and I would like to thank my professor Sendil Kumar sir and my madam Dr. Tilagam, who has taken classes from the first B Pharm onwards. Okay, when I entered in 1992. Okay, so I am a first batch student who joined this JKKMMRF in 1992. Okay. So now I have seen a big transformation of this institution. Okay, at that time we don't have anything, no buildings, nothing else. Okay, at that time we are only small, 4 or 5 classes and a few faculties. Okay, in that aspect, in that thing, we have followed the guidance of Sandil sir, Telagam madam and lot of professors. Okay, we are the first batch, and our first batch students all are in high level positions. Okay, really very happy that they first seeded. Now itself started the cognosy. Okay, so first seeding is happened here only for us. Okay, so on that aspect, really I have to thanks to JKKMMRF for giving this opportunity and very good transformation and transforming us. Okay, so now this topic is related. Okay, transformation, differentiation, everything is coming under this stem cell regeneration capacity. As Dr. Ravi Kumar, he has mentioned a word, regeneration. Okay, I hope you will be knowing a lot of degenerative disorders. Okay, as madam has taken about the rheumatoid arthritis, okay, osteoarthritis, okay, Alzheimer's disease, maximum all are degenerative disorders. Okay, so for that degenerative disorders, we are
having a lot of drugs. Okay, so that drugs, what it will do, it will act on the anti-inflammation or something. It will act on the symptomatic pathway and it will release, it will block the some mechanisms like that. But you cannot go for total regeneration of that particular tissue. Okay, so you can create a new organ. Okay, so creation of a new organ is not a simple process. Okay, all of you knowing that during the pregnancy, okay, so they are going for scanning whether the zygote is formed and how about the developmental activities and how about the formation of the limbs, how about the formation of the brain, and within a 10 months, okay, within a 10 months, listen here, within a 10 months you will be getting a baby with functioning of all organs perfectly. Okay, so within a 10 months, how much of the things is happening? Okay, so eye formation, heart pumping, okay, all type of sensory things, okay, it is formed. A whole body is formed within a 10 months. So how it is forming and what all the rapid things are happening in the during the pregnancy and after the delivery. Okay, so those things only are coming under the stem cell differentiation. Okay, so now we are using some advanced technology of using the stem cells and formation of the organs. So now on this aspect, how we can correlate the natural products. Okay, how we can correlate the natural products for this differentiation and regeneration. Okay, so always we should not be against the nature. That's why only I'll be telling to my cognosy department people, madam, we are not against the nature. If you are skipping the nature, okay, so formation of baby itself is a big nature. Okay, so if there is any small mechanism, if it is not happen, means what happens? Total loss. Okay, total body, everything will be gone. So the nature is nothing but degeneration only. If you are getting aged, okay, if you are getting aged, what happens? Totally degeneration. Okay, so aged, what type of degeneration is happening for the aged people? It is coming Alzheimer's disease, Parkinson's disease, arthritis. Okay, so all type of cell mechanisms will be lost. Okay, so we have to go towards the nature and we have to stabilize the degeneration process. That is the meaning we can say it as a regenerative medicine. Okay, so when you are applying a technology for the regeneration process, we can say it as a tissue engineering. Okay, if you are applying a technology by using the biological system and also by using the natural products, you can say that is known as the tissue engineering. So my topic involved is plant-based scaffold for stem cell differentiation and regenerative medicine focused towards the tissue engineering process. Okay, so totally you are doing all type of work. Okay, you can do all type of work, whether that product is going to the market or not, that plays a major role. Okay, if you are preparing a product, that product should be used for the hello. So that product should work, that product should be used for the society. Okay, so if that product is not used for the society, then what is the use of doing all other works? Okay, so coming to that aspect, the product is very, very important. So we are using some advanced technology for creation of the products. Okay. This is okay. I would like to ask one question. I hope everyone went to hotel, right? Hotel எல்லாருமே போயிருப்பீங்க, ரைட்டா? ஹோட்டல் கிளீனர் வந்து அந்த டேபிளை வந்து க்ளீன் பண்ணணுன்னா என்ன பண்ணுவார்? எஸ், okay. So just I want to ask a question, okay? The hotel cleaner will be using a sponge, right? He is using even that sponge is available in your house also, okay? Just you are taking that sponge and you are cleaning it. It is having a two property, okay? So one is that sponge will absorb the water, it will hold it, okay? And whenever you are pressing, that wastage will be going outside, right? So here that sponge, you can say that sponge, you can say it as a scaffold, okay? So if you are taking that sponge, you are adding some drugs or any other molecules, and you are taking that sponge and you are implanting to the human body, means there it will form an organ. Okay, just for example, I am telling an organ for your understanding. Okay, generally we can say it as a formation of that particular tissue. Okay, so now here the topic which given to me is we have to go towards the natural things. Okay, so how these natural products can be used for the tissue regeneration and can be involved in the regenerative medicine. Okay, so now what they are doing is they are using the scaffolds. Okay, so two aspects you can make it. Okay, so one is you can prepare these scaffolds by using the plant materials. Okay, second one is with that scaffolds you can add some drugs for regeneration purpose. So two aspect I am going to take it. Understand? So now what we can see is you can take the plants. In that plants, what are all the things? You can use the plant extracts, you can use the plant proteins, you can use the plant constituents, maybe polysaccharides and all. Okay. So now this diagram, just for your simplified things, just I want to understand, I want make you to understand. Here you can see what are all the things you can see. You can see an apple, you can see a carrot, you can see a broccoli, okay, you can see an onion, you can see some leafy vegetables. Okay, so these things is having a property and having the application for the regeneration process. Okay, regeneration process of what? Heart, you can go for muscle, you can go for bone, and you can go for skin regeneration process. Okay, so how you think? Generally what they will say, just if you take an apple, it will produce some antioxidants. If you take some leafy vegetables, it is very, very useful like that. So that is totally different. Okay, so that one you are taking it in a internal way. Okay, but once if it is coming to the applying the technology, and if you are applying for the other things, means you have to go for the advanced things. Okay, you can develop the advanced things. Okay, when I hope when you will be knowing about the phones. When I am studying, we are not having any phones. Okay, we are having only the landline. Then afterwards we got a small phone. Then afterwards, now another Android phones, iPhones. Now it is going in a advanced way, but we have adapted to that environment. Okay, so like that, now you are doing final year, first year or final year, but when you are coming, okay, when you are coming to the, after completion of your B Pharm and M Pharm, you will be involved in the technology based works. Okay, you will be involved in the technology based research works. Okay, so there you have to go for this type of scaffold system for regeneration purpose. Okay, so nowadays we are taking a tablets and other things and all, but now after in the advanced things you will be going for cell therapies. Okay, so now I'll be telling you how we can use these apples. Okay, you will be knowing what is an apple. Okay, you will be knowing what is a carrot. You will be knowing what is a thing. So only thing is, apple structure is totally different. Okay, leaf structure is totally different. Okay, carrot structure is totally different. So architecture, what is that architecture? Architecture means that is a structure, what it presents, how the cells are built, and what is the extracellular matrix. Okay, so based on that, you can create a specific thing for organ regeneration. Okay, for example, if you take a bone, okay, so bone is very hard. If you take a heart, okay, so heart is very hard tissue, no? Comparing to other things, heart is a very, very hard tissue. You cannot slice the heart. You cannot cut the heart very easily. Okay, but it is a very hard tissue. It has to pump the blood for throughout your life. Okay, it is very, very hard. Okay, and if you take a skin, okay, it is totally, our body is totally made up of skin. If you see a 3rd degree burns, okay, so the patient will be live for 3rd degree burns for at least 2 or 3 days, but 4th day automatically the patient will die. But up to 3 days you can observe the patient will be in the normal state and he will talk everything. But in the 4th day he will be losing because there is no skin regeneration, angiogenesis stopped, everything will be stopped. Okay, so all type of organ regeneration plays a significant role for in all type of degenerative disorders. Okay, so now this is a very simple example, just I am telling you, only with this type of whatever the things you are having itself, you can make some technology and you can go for tissue engineering purpose. Can you able to understand, no, what I am telling? Are you following, no? Okay, I am just giving an example. Okay, in that example, you can use the spina cleaves for vascular network. Okay, spina cleaves. If you take a spina cleaves, you can see the stem and other things, how it is formed. You can take that one and you can go for the vascular network. Okay, and apples. Apples can be used for formation of the bones, osteoblast. Okay, osteogenesis we can say, osteoblast. And green onions can be used for skeletal muscles, and bamboos and lotus for the bone tissue engineering. Okay, so I hope it is somewhat what sir is telling is simply other things, and you can go for formation of the specific things. Okay, you will be thinking, okay, so there are having a lot of mechanisms. Okay, you need a more skill, you need a more ideas. Okay, everything is needed. So I told you what is a scaffold. Scaffold is nothing but it is a extracellular matrix. Okay, it is a extracellular matrix which supports the growth of the cells. Okay, So first example what I told, the cleaning person, if he is having a sponge, you can take that sponge, okay? You can cut that sponge, and with that sponge you can add some cells, you can add some other constituents, and if you keep means, it will form a exact whatever the things you want to target, that cells will be formed. Okay, it is a very simple example, just for your understanding I am telling you. Okay, it is a extracellular matrix support. Understand, it's a extracellular matrix support. So in what way? Already I told you 2 points. Okay, so one is you can use the plant products as the extracellular matrix, or you can use the extracellular matrix along with some constituents to make it for the regeneration purpose. Okay, so with the 2 concepts only I am taking this, I'm going for these presentations. Okay, so first one is decellularization. Okay, so to get the extracellular matrix, you have to remove all the cells. Okay, you have to remove all the cells. இப்போ லீஃப் எடுத்துக்கிட்டீங்க அப்படின்னா அந்த லீஃப்ல இருந்து என்ன பண்ணலாம், செல்ஸை மட்டும் ரிமூவ் பண்ணிட்டோம்னா எப்படி இருக்கும், ஒயிட் கலரில் அழகாக வெறும் இது மட்டும் இருக்கும். புரியுதா? ஸோ லைக் தட் யூ கேன் remove whatever the even stem like that and all. So once if you are removing it, it will form the extracellular matrix. Okay, so next one is functionalization. Okay, so after removing that that cells, you can make it into a functionalization by attaching some other molecules, and you can make them the cells, mammalian cells, to develop into that vegetative cells, vegetative matter. Okay, so like that if you add some other cells, and you can create a other structures. Okay, and that is the recellularizations. Okay, if you add a cardiomyocytes, that thing you can use it for the cardiomyocyte regeneration. That one can be used for the, what we can say, you can go for you can use it for this cardiac alignments. Okay, so there, which plays a major role. So you need a lot of technique to create a simple things into a technology matters. Okay, so that is very, very important. Even you will be having some ideas. Okay, you can see small ideas will be having a biggest applications. Okay, which is having a very excellent applications. Okay, so like that the idea is very small, but it is having a highest applications. Okay, so how we can use a small, very, very available natural products to going for a highest important regenerative purpose. Okay, that is one point I have discussed. Okay, and another one, okay, already I told you, stem cells plays a major role for the regeneration purpose. Okay, now if you think, if a person is sitting near, okay, if he is interested, okay, if நார்மலாக நல்லா படிக்கிற பசங்களோட சேர்ந்தா என்ன பண்ணுவோம், இவங்களும் படிக்க ஸ்டார்ட் பண்ணிடுவாங்க. சரியா? படிக்காதவங்களோட சேர்ந்தோம்னா படிக்காமையே போயிடுவாங்க. புரியுதா? அதே மாதிரி தான் இங்கேயும். புரியுதா? அந்தந்த ஸ்டெம் செல்ஸ்க்கு என்னென்ன growth factor, what growth factor we are giving, according to that it will be get differentiated. Okay, so for the differentiation process you have to add the growth factors. Okay, you have to add. For example, if I am having a stem cell, if I am adding a brain derived growth factor, if I add a brain derived growth factor, means automatically that stem cell will be converted into a brain cells. Okay, if I add a hepatocyte growth factor, that stem cell will be differentiated into a hepatocytes. Okay, so like that the cells can be changed according to the growth factor. That's only we can say it as a differentiation process. Okay. So how we can make it? How we can make a differentiation? And what is the role of the natural products? Okay. So previously, now it has taken me a very easy, because madam has given a lot of information about the signaling mechanisms. Okay. So in that signaling mechanisms, you have to modify that signaling mechanisms by using the natural products. Okay. So you have to block these specific signaling mechanisms. By that, you can make it for the exact differentiation process. Okay. For example, I am asking you, what's the difference between your ear and your just you can take a nose. Okay. The nose is made up of, these things is made up of a cartilage. But if you go enter inside, it is made up of bone. Okay. So there is some transition between the cartilage and also the bone. Okay. There should have some maintenance. Okay. If you are not giving the exact mechanism, means the cartilage will be converted into a bone, or bone may be converted into a cartilage. Okay. So it will become a very soft tissue. Bone will become a very soft. Okay. If the cartilage becoming a very hard, what happens? If your ear is becoming a very hard, what happens? So like that, there are several mechanisms are involved in that to modify the exact signaling mechanisms and focusing towards the tissue regeneration process. Okay. So first one is differentiation and regeneration. Okay. So how we can make it? How we can modify these things? Okay. So here you can use some compounds. You can use some extracts. Okay. So that extracts can act on that signaling mechanism and it will go for the particular regeneration process. நீங்க prepare பண்ற ஏகப்பட்ட extractsலாம் இருக்கும். Okay. Even final years they are doing a lot of extracts. Okay. So you are doing several activity. Okay. In that activity you will be giving a exact results and you will be getting a very positive approaches. But what is the exact mechanism? That is very important. If you know that exact mechanism, you can make that one for the technology process and you can create a product, and that product can be used for the market. Okay. So I'll be showing what is the value of the tissue engineering nowadays. So like that. So scaffolds, cells, signaling molecules, antisense. Antisense I'll be explaining afterwards. Okay, all these things it can make it for the going towards the tissue engineering and the regeneration. So multidisciplinary research work is very important. Okay. So I need a help from the pharmacognosy, I need a help from the pharmaceutics, I need a help from the pharmaceutical chemistry, I need help from pharmacology. So all persons, all multidisciplinary activities are needed to get one product. Okay. It's not like a individual person can make a product. Okay. So all members' ideas are very important. So don't think that only one thing is very important. Okay. So that's only I am telling you. As I told previously, what I told is scaffolds can be prepared by using the plant material itself. Okay. It's a very simple process. என்ன இருக்கிறத எடுத்து காய வச்சு ஏதோ ஒன்று பண்ணி நாம் என்ன பண்ணலாம், அந்த செல்ஸ் எல்லாத்தையும் ரிமூவ் பண்ணோம்னா அதுவே ஒரு ஸ்கேஃபோல்டாக மாறிடும். புரியுதா? அந்த ஸ்கேஃபோல்டில் உங்களுக்கு தேவையான செல்ஸை போட்டு இன்குபேட் பண்ணி அதை பண்ணிங்கன்னா அதுவே என்ன ஆகும், டிஷ்யூவாக ஃபார்ம் ஆகிடும். ஓகே. ஸோ லைக் தட். அண்டர்ஸ்டாண்ட்? So this is a thing which I want to make it. Okay. So now the application for the tissue engineering is you are adding all these things and you are making a tissue engineering scaffold, and that one can be used as a tissue engineering product. Okay. So now it is somewhat just I am entering inside. You can see that different types of fruits, vegetables and everything. Okay, they are taking it somewhat advanced level. they are making that one into a scaffold, and with that scaffold they are adding some molecules and they will be checking for the regeneration process. Okay. So each organ is having some specific architecture. Okay. Brain is having a different architecture. Bone is having a different architecture. Heart is having a different architecture. Okay. Porosity is different. Cell function is different. Okay. So like that you have to identify the architecture. Based on that you can select from the natural products, and that one you can be use it. Okay. So by that it can be produce products. Okay. So there are a lot of things I don't want to explain. I think this and all are the molecular studies you can do, and you can make it, so you can substantiate, and by that you can make it. Okay. So now another technology is whatever the things you are getting. For example, if you are taking aloe vera is having some gelling property. You can take that gel and you can make it a nanofiber, and that nanofiber you can make it according to the architecture. That is a very, very excellent technique. Okay. So polymer chemistry are doing like this type of things. Okay. So like that you can make it. Okay. So like that you can do, and we are working. Okay, as we are working on these three areas, neuroregeneration, skin, cartilage, and the cardiomyoblast regeneration proper, and we have done a lot of work, and it has got a very excellent publications in the impact factor journals. Okay, so I don't want to explain all these things, you can see. Okay. So we'll be showing a last slide. This thing we are working on, this signaling mechanisms to modify the regeneration purpose. Okay. So this is one of our scaffold we have prepared. In that you can see the cells has got attached, and it has got a very excellent wound healing property, and got published in the International Journal of Lower Extremity Wounds. Okay. So we are okay, so this nanoparticle incorporations and modification of the nanoparticles, okay, all these things you can make it. So this is a very simple process. Okay, just you see this slide. Okay, in this slide it is very, very easy. You are preparing a scaffold, that is the 3D porous scaffold. You are adding the cells, and you are taking that one for the implantation. That one it is giving the regeneration purpose. Okay, so that is known as the 3D printing. If you want to go for 4D printing, 5D, 6D also you can make it. For example, according to the size, according to the shape, your implant has to fit to that particular area. Okay. For example, tracheostomy they are doing. If you are cutting the tracheostomy, there are a lot of cells are there, angiogenesis. Okay, a lot of things are there. The angiogenesis has to be carried out. So like that you have to 4 or 5 different layers you have to make it, and that one you have to implant it. That is the 4D technology. Now it is in advanced technology, IITs they are doing. Okay, so like that lot of work is going on towards the tissue engineering purpose. Okay, so see, you can see the fennel seed extract. Okay, only a fennel seed extract is having the differentiation in the stem cells, mesenchymal stem cells for the bone formations. Okay, so like that lot of things we have carried out, and this is another one which we have patented, and it is having an excellent wound healing property. Okay, PVA, you can combine the synthetic and also the natural polymers isolated from the plant, and you can do the things. Okay, so like that lot of things which has got published. Okay, so next generation plant-based scaffolds. Okay, plant-based scaffolds has the potential to revolutionize the biomedical applications. So how? 2 points. One is you can create a scaffold from the plant, Another one, with that scaffold, you can add the plant constituents going for regeneration purpose. 2 points, okay? If you understand this concept, you can do a lot of wonder things. You can create a lot of regeneration purpose, and that scaffold is having more value. And if you are making it in a simplified way, it is a cost effective and it is having high market value. Okay, thank you.
An excellent session which has taken us a lead for a scaffold formulation. I hope the audience would have utilized it. This could have been a lead for you in your UG or a PG project. Kindly take into account of the efforts which are being taken by the resource persons. It is out of their interest. They are over here to share their experience. You have got a chance. It is not possible for you to move all around, either to JSS or to Worlds or to Pondicherry or, right, Kerala, Agalya. But the resource persons are over here to pass their messages, their efforts in the research for the future generations. The future generations don't think that coming to a seminar just to sit and chat. Please listen, take home messages. I am sure this will be a lead. If you make use of it, surely it will be good for you. You can take a lead. So here, Sir has taken a lead in the scaffold. Right from the basic example of a sponge, from a sponge how we can make a scaffold. He has taken various appropriate examples of plant tissues and tissue engineering. What is it? Definition. What are the goals? What are the applications? And the lead, the best example of his publication. So now the session is open for any questions?
Sir, you presented nicely and you said development of scaffold from plant-based, right? And after acellularization, decellularization, we are directly going for functionalization. Functional modifications. How do you ensure, because it's a natural polymer, how do you ensure the mechanical integrity? Generally, we do for scaffold preparation from natural polymers like collagen or chitosan, we do cross-link. Is there any cross-linking for natural is needed or what?
See, normally, once after decellularization, you can observe the cellulose moieties. So that cellulose moieties, sometimes OH groups or COH groups. So that one will be adding the NH2 groups to attachment of the cells. So this attachment, for example, if you take a cells is having the receptors, that one it will be get attachment and it will go for the migration process. So for that only this decellularization, and you are going for other chemical modifications and you will be attaching those ones. So by that it will be go for attachments and migrations, and afterwards to that decellularized scaffolds you can add the constituents for the formation, that gel constituents. So that one it will differentiate the cells for the formation of that particular thing. Okay. So two aspects, sir. One is you can modify the scaffolds. You can modify the scaffolds. With that modified scaffolds, you can add the cells. One aspect. Another aspect is you can add the constituents for the any other scaffolds, plant constituents, to target the signaling mechanisms to accelerate the differentiation and regeneration process. So that is the two aspects we can make it. So now it is a recent developments is going on for the modification, but without the modification itself, they have got a several excellent results they are doing. Now we need to modify all the things.
One more thing is, in biological polymers,
SEM studies, after the decellularization process, you can observe the pore size. See, pore size is very, very important according to the architecture. See, suppose if you are taking a leaf decellularization, will be getting the different pore size. Suppose if you are taking a stem, that getting different pore size. Okay, so according to the pore size, will be getting the cells to attachments. Okay. See, pore size is totally different for each architecture. That's only I told. For according to the architecture, the pore size only plays a major role. In that, we will be seeding the cells and going for the differentiation process.
Thank you, sir.
So I'd like to conclude the session where the transformation of tissue engineering into an regenerative medicine, that is highly possible. So on behalf of the organizing committee, I thank the resource person for taking his efforts to bring out the importance of scaffold. Thank you, sir.
Thank you, sir. Now I request Dr. P. Kalachelvi ma'am, HOD Department of Pharmaceutical Analysis, to honor our speaker, Dr. T. S. Shanmugarajan, sir. I request Executive Member, APTA Member, Dr. P. Satish Kumar, sir, to honor our Chairperson, Dr. C.S. Kandasamy, sir.
Thank you, sirs. Let's have a lunch break for 30 minutes. The lunch will be arranged for students near to girls' hostel in ground floor. Students should assemble at 2:30 p.m.